Okay, so we'll start with particle size analysis. So over here, it's particle size analysis is when you have like a powder mix and you want to see how what kind of particles or what size particles you have. Just a minute. Yeah, so it shows which size of particles you have. So the first one a method that you use is the sieve method, where the lowest sieve diameter is usually 45 micrometers and the highest is 1,000 micrometers. So in this one, everybody knows what a sieve is. So what in the process they usually have, like, the sieves are stacked, like 6 to 8 sieves which have different the mesh size is different. And like we've seen, we had done, I think, a practical with Muniendo, where we saw like each sieve has the mesh number written on the sieve. So it's usually that the sieve size with the highest, with the biggest diameter is usually at the top, and the lowest or the smallest is at the bottom. So that's one way to, and then you pour all your powder mix, and then you start that sieve at like where it like this vibration, and then it separates the particle size and you get your different particle size. Then another one that is used is called the air jet sieving, where only individual sieves are used, while in the previous one we saw that it was many sieves. So over here, what the air does is that it basically prevents the mesh from being blocked by the oversized particles, and it's more efficient and reproducible, because once the mesh is blocked, then even the smaller particles will not be able to go through the mesh. So when you have this air jet sieving, it's more efficient and reproducible because it will separate out all the particles. The next is microscopic methods. So the 3 types of microscopy: light microscopy, where the sample is usually dispersed on a microscope slide. Second is scanning electron microscopy, where the sample is usually fixed on aluminum stubs. The third one is the transmission electron microscopy, where the sample is often it's set on resin. So the first light microscopy is like the simple one that we've also we've seen in labs, but the scanning and electron microscopy and transmission electron microscopy is now on the higher side, and it can show you a particle size of like a very small particle size, like you can see in this diagram over here, where TEM is like from 0.001 to whatever. So it will give you a more accurate reading, and it will also show you the smallest particles that are in that powder mix. So for light microscopy, it's usually carried out in a 2-dimensional images, so you can project those images and see how the size of the particle. And then you can use so basically you can use a projection screen with the screen distances related to the particle dimensions, using a reticule. And we've done this with in microbiology, where we can the graticule is usually that black line, and you can see the size of the particles. Then I'm just going through like the important ones. Yeah, so then there are also like semi-automated methods where the microscope analysis uses some form of pre-calibrated variable distance to split particles into different size ranges. So like you can see over here, the particle size is projected on a photomicroscope, and then you can vary this aperture of the iris to see which what fits with that particle and determine your particle size. The second one is a double prism arrangement that is mounted on a light microscope eyepiece. Then the third one is the sedimentation methods, where you have centrifugal sedimentation and gravitational. So when with samples for this one, when you're doing the sedimentation method and the powder is not uniformly dispersed in a fluid, it can be introduced as a thin layer on the surface of the liquid. That's one case. Second, if the powder is hydrophobic, then you can add a dispersing agent to aid wetting. That's the second case. And third case is where the powder is soluble in water, but you have to put it in a non-aqueous liquid to carry out the analysis, or you can even put it in gas. So those are the three ways you can prepare your samples when you're doing sedimentation. Firstly, when the powder is not uniformly dispersed, you introduce it as a thin layer on the surface of a liquid. Second, the powder is hydrophobic, then you add a dispersing agent. And third, if the powder is soluble in water, it will be necessary to put it in a non-aqueous liquid, or you can put it in gas. So for sedimentation, there are two types of methods that are used, or two measurements. The first one is retention zone, or let's start with non-retention zone. Non-retention zone, where you use a pipette method. In this method, known volumes of the suspension are drawn off and the concentration differences are measured with respect to time. So you have a suspension, you leave it, and you insert a pipette at a certain, until a certain like length, and you leave the pipette over there. Then after like 1 minute, you take that sample out, you look at the concentration of the particles. Again, after another like 2 minutes or so, you remove it from that same place, and you look at the concentration. So in theory, or even in a practical, we would be able to see that the concentration of the particles would decrease because of sedimentation. Then the second type is the retention zone, where sediment size analysis is done using the Stokes law to quantify the particle size. And over here, it's usually the sedimentation balance that's used. And what's done over here is that the amount of sedimented particles falling on a balance band is suspended in the fluid is recorded. And over here, it's the weight of the sediment that is recorded. In the previous one with the pipette method is where the concentration of the suspension is what's recorded with time. So one of the limitations for these sedimentation methods is that the diameter is a diameter of approximately like 5 micrometer. Just a minute. Yeah, sorry. If the diameter of the particle is more approximately 5 micrometers, then the sedimentation can be prolonged because of interference from like diffusion or like Brownian motion. Then the fourth one is electrical conductivity method, where in electrical so a particle suspension is usually like put in a a container or whatever, and then you the electrodes are put. So now over here, what they are looking for is a change in electrical signal that occurs when a particle momentarily occupies an orifice. So over here, you have an orifice. So once that particle goes through that orifice and displaces its own volume of the electrolyte, there'll be a change in electrical signal. Then this change in electrical signal will now be processed electronically, and you can see the size distribution of the particle. Then another type is now attenuation of a light beam by particles drawn through the sensing zone. Then I think this is the last one, the laser light scattering method. So this one has like two of them. So the first one is the Fraunhofer diffraction, where particles that are much larger than the wavelength of light, when they interact with light, they will scatter the light in different directions, and a small change in angle will occur. So that is what the diffraction will occur. So if it's if the particle size is larger than the wavelength of light, once it hits the light, it will diffract the light. So you can see, you can determine the particle size from that. And the light emitted is usually by a helium neon laser. Yeah. Then the second one is now the photocorrelation spectroscopy, and this uses now Brownian motion to measure particle size. So what it does that these particles, they go through this Brownian motion, and it monitors the rate of change of scattered light during the diffusion. Again, it's the same principle of scattering light, but now this is it monitors the rate of change of scattered light during diffusion. Over here, it was a change in the angle because of the diffraction. So that is it for particle size analysis. Do you guys have any questions? I'll go to the next one then. Any questions, or I go to the next one?
No questions. No questions from me, unless someone has a question. But okay, one question about this particle size analysis. Have you seen it being covered a lot in the past papers?
Not really. That's why I went, like, I did like a brief overview. Like it can
just
ask
an
you.
overview. Oh,
Yeah.
nice.
So instead of going into like that details, I just went like whatever I'll be teaching today, it will be just like an overview, because since it's multiple choice questions, they won't ask us for the method of how it's done. It's mostly going to be things that you can pick out. So that's what I've done. So it's just like an overview. So let me just let's just do an overview again. So particle size analysis, you have the sieve methods, where you use a sieve to separate out the different particle size. Next is microscopic methods. Now that you can see the particles itself from either light microscopy, scanning electron microscopy, or transmission electron microscopy. Next is the sedimentation method, where you use the retention zone where the sedimentation balance is used, and the non-retention zone where the pipette method is used. Then you have the electrical conductivity method now, where you measure the change in electrical signal when a particle occupies the orifice. And then the last one is the laser light scattering methods, which has the Fraunhofer diffraction and the photon correlation spectroscopy. So we just go next. Just give me a few minutes, I'm coming. Just a minute. Okay, so the next one is, you guys can see my screen, right?
Yeah, we can see your screen.
Okay, so the next one is particle size reduction. So when we're talking about particle size reduction, we have to talk about the size grading efficiency, which is related to the particle and fluid properties and the grading method that is used. So particles are usually separated into oversize and undersize. So when you need to calculate the functions of the mass flow rates of the feed material, you have to add the oversized product and the undersized product, because even when you're reducing particles, it's not that you're going to get the same amount of particles. You're going to get, the same size of particles. You'll get oversized and undersized, or in the middle. So when the process is 100%, all oversized materials will end up in the oversized stream, and all the undersized material will end up in the undersized product stream. But that does not happen in reality. So there's always incomplete size separation. Again, I'm just giving an overview. Yeah, so now size grading just yeah. So the first one is the agitation method, where size separation is achieved by electrically induced oscillations or mechanically induced vibration of sieve meshes. Again, you're using sieves over here, where you oscillate them or like to induce vibration, and that will reduce the particle size because it has to go through that the mesh size. Yeah, so that's the first one when we're talking about sieving. So that's the first. In sieving, you have agitation methods. Second is the brushing methods. Now, a brush is just used to now prevent the apertures being blocked. In the previous presentation, we saw that an air jet was used to prevent the apertures from being blocked. Then the third one is the centrifugal methods, where the particles are thrown outwards onto a vertical cylindrical sieve under the action of high-speed rotor inside the cylinder. So when there's certain standards for powders based on sieving. So when you want to have when you need viscous particles, then you use a sieve diameter that is around 1700 micrometers, and then like you can see for very fine is 125. And yeah, so that was the first one, is size grading by sieving, where you have agitation methods, brushing, where just a brush is used to prevent the apertures from being blocked, and centrifugal methods. Second one is now sedimentation methods, where it utilizes the difference in settling velocities of particles with different diameters. We know that the bigger the particle or the heavier the particle, the more it will easily sediment compared to now the smaller or fine particles. So this method over here, this whole thing, is what I explained in the pipette method in the previous one, where you insert a pipette into a flask or whatever at a certain position, and then you pipette out a certain amount of the suspension at different times, if it's like 1 minute, 2 minutes, or whatever, and then you see the concentration of the particles or see how many particles are there. What you expect is with time, the particles will now reduce because sedimentation has occurred. Now that's one of the simplest way. Then now there's other methods where like the suspension is put into this inlet over here, and then it goes, this is just filled with fluid, and you expect that the bigger particles will go towards the first outlet, compared to the smaller particles which will go to the furthest outlet. Then this is another one. Just a minute, guys. Sorry. Sorry about that. Yeah, so in this one, the suspension is put through the suspension is put through this inlet, and you expect that the particles with the biggest or the heaviest will settle down first, compared to the smaller particles which will now go or settle towards the outlet. So the particles are acted upon by two forces, the fluid flow and the centrifugal forces. And then the third one is elutration methods. This is like opposite of sedimentation. In sedimentation, the fluid flow and the particle flow is the same direction, but in this method, the fluid flow is opposite direction to the sedimentation movement. the particles move vertically downwards while the fluid moves vertically upwards. And because in this case, the particles are divided into different size fractions depending on the velocity of the fluid. So in practice, we see that when you do this experiment in like a tube, the highest velocity in the tube is usually at the center, and the lowest velocity is usually on the sides of the of the tube. So what happens is that the size of the particles will be graded depending, the largest particles will be at the center where it has the highest velocity, and the smallest will be outside where it has the lowest velocity. Then another method over here is that the suspension is let into from this one, and then it goes from 1 and then 2 and then 3, but then the diameter of those tubes increases. So as the diameter increases, the fluid velocity decreases, and therefore the decreasing size. So you expect that in tube 1 you'll have the biggest particles compared to tube 3, because as the velocity decreases, the more you'll get more of this smaller particles. Then the last one is the cyclone method, where particles sediment out in a helical gas or liquid stream. So like you can see over here, the particles are let into from this place, and then what happens is that the coarser particles usually go into the particle outlet, while the smaller particles will like they'll be more entrained into the fluid stream and just get out from this vortex finder. So from over here, you'll see the bigger particles or the coarser particles, and from the fluid outlet from the vortex finder, you'll see the finer particles. So let's do a recap. Size grading methods. First one, sieving. Sieving has agitation. It has brushing methods and centrifugal. The standards that you can use that are based on sieving, if you want to have like fine particles, then you use a sieve size of 180 micrometers, or that you can see over here. Second is through sedimentation methods or through fluid classification, where you can use the pipette method. You can use this method over here, where you'll see coarser particles in the first outlet and the finer particles in the second outlet. For this one, you'll see that the coarser particles will sediment first compared to the finer particles, which will be at the end of this maze thing. Then the third one is the electrician method, which is opposite of sedimentation, where the fluid flows in the opposite direction of the sedimentation method. And when we're looking at it in practice, when you're using like a tube for this method, the highest velocity is in the center, where you'll see the largest particles, and on the sides of the tube it will have the lowest velocity, where you'll see the finer or the smallest particles. You can also use this, where in the first outlet you'll have the coarser particles or the larger particles, and once it moves further into this other tubes, it will have or the columns, you'll see more of the finer particles, because as the diameter of the column increases, the velocity decreases, and therefore you'll have more finer particles. Then the last one is the cyclone method, where the coarser particles will be let out from this particle outlet at the bottom, while the finer particles will just be entrained in the fluid stream, and it will go towards the vortex finder. So that is it for size grading. Any questions?
No questions. No questions.
Okay, so then we'll just go to the next one, which is now particle size reduction. So the previous one was particle, that was size grading, and this one is particle size reduction. Sorry about that mistake. So what happens in particle size reduction is that you have to like propagate a crack into a particle, and like through a stress, like you apply stress to that particle and a crack will propagate. And the crack, like it's usually large enough to cause like bond rupture and thus like propagate a crack. So when you want to reduce the particle size, you have to look at the properties of that material. So if you like, if some particles that are not brittle enough, or if they have like plastic flow, then a crack will not be, you cannot be able to reduce the size of that particle, such as like rubber, because if it has plastic flow, the molecules just they just slip over one another. So yeah, so rubber is one of the examples that has like plastic flow. Then when we talk about surface hardness, we need to see that these harder materials are more difficult to reduce for reduction, and they can even damage now the parts of the machine when you're trying to reduce the size. So in such a case, yeah, okay, and then we said that that it has, if it has like an elastic component like this rubber, what can you do to make it so that you can reduce the size? So what you can do is that you reduce the temperature of that material below the glass transition material, making that material go from plastic to brittle, and it will be easier to propagate the crack. Okay, and then energy requirements for size reduction process is that not all the energy used is for to propagate the crack, but there's some that is lost in like elastic deformation of particles, particle deformation of particles without a fracture, deformation to initiate cracks that cause a fracture, deformation of particle of the metal machine parts. Like I said, that some of these materials are like they're hard enough or they're brittle and they can damage the machine parts. Then you have interparticulate friction. Then you have particle machine wall friction, heat, sound, and vibration. So not everything is gone into propagating the crack, but some energy is also used for these other processes. So there's some like 3 theories that is used for this energy. So the first one is the Rittinger's hypothesis, where the energy is proportional to the new surface area that is produced after the crack. So the first one, Rittinger's hypothesis, is where energy is proportional to the new surface area that is produced, the new surface area. Second one is the Kick's theory, where energy is proportional to the ratio of the change in size. So you know you're going to reduce the size, the ratio of the size change in size will be the previous size which would be larger compared to the new size. So Kick's theory, proportional to the ratio of the change in size. Third one is the Bond's theory, which is proportional to the new crack length that is produced. Then this Walker's theory is now a combination of all three theories, mostly Rittinger's and Kick's theory, and in some cases with Bond theory. So in this equation, we can see this n over here, which is like a constant kind of. So when n is equal to 1, it will be Kick's theory. When n is equal to 2, it will be Rittinger's theory. And when n is 3 over 2, then it will be Bond's theory. Yeah. So when it's 1, it's Kick's theory. When it's 2, it's Rittinger's Rittinger type solution results. And when it's 3 over 2, then it's Bond's theory. Yeah, and then when we're looking at this equation where we have to find n, over here. So when the particle size increases, this f function of, I think it's function of this diameter, I'm not too sure, but yeah, it tends to be 1, and as the size reduces, it tends to be 2. So if this FD is 1, then it's Kick's, the n will be Kick's theory, and when n is 2, or FD is 2, when the size reduces, then it's Rittinger's theory. So for this one, influence of size reduction on size distribution, when you're reducing the size again, you don't expect to have all same sizes. You'll have like a range. So when you plot a graph, it will usually have this kind of shape, where you'll see that the extremes are on a lower side compared to the one that is in the middle, which is higher in frequency. Yeah. Now, when you're coming to size reduction methods, first is the cutting method, where you use the cutter mill. Now, I'm not going to explain how the whole machine works. I think we need to remember what kind of a technique or equipment is used. So for cutting method, you use the cutter mill, which has now this different types of knives, and it can reduce the size. Second one is compression methods, where on a smaller scale you use like this mortar and pestle, but when you're going into a larger scale, this end runner and edge runner mills are used. So an example over here, you can see where the sample is fed into this, and these hammers like cause compression of the particles, and which reduces the size of the particles. Next is the impact method, where the hammer mill is used. I don't know if they've not given a picture of that. But I feel it's similar to this one because this has the hammers also. So impact methods, hammer mill, but you can also use this vibration milling, which has this different kinds of steel balls. Yeah. Then attrition method, where roller mills are used, and this is mostly when you're making suspension. Then you also have a combined of picture, yeah, over here. So yeah, so this is usually, can you guys hear me?
Yeah, we can hear you. Yeah.
Okay. Yeah, so this ball mill is usually used, and these are all, I think they're steel or porcelain, and it is spun in a certain velocity, but you have to take care that if it's a high angular velocity, then it will cause this centrifugal force, and it will just, like, that centrifugal thing will happen, and there'll be no size reduction. So you have to take care, uh like, what is the angular velocity that you need for size reduction to occur. So when it's in a case like this, where the balls are like at a high level and then they drop, so it causes an impact on the particle, causing size reduction. Then so all these, where is it? Yeah. Yeah, then the second is this fluid energy milling. So what happens over here is there is yeah, the particle is reduced by impaction and attrition. And usually you have this fluid inlets, where a fluid, which is usually air, is injected at a high pressure, and the solid is put through this inlet, and like high velocity of the air gives rise to a zone of like turbulence, and then the solid particles collide with each other, or with the walls of this equipment, and causes like, yeah, and it causes size reduction. Then selection of powder reduction method is usually, it usually depends on like two things, where you have to see what kind of size reduction or the degree of size reduction that you want, and of course the cost of the size reduction, because as the particle, yeah, the cost of the equipment also. So that is it for particle size reduction. We can just do an overview. First is cutting methods. Cutter mill is used. Second, compression method, where you have, on a smaller scale, you have mortar and pestle. On a larger scale, you have this end runner and edge runner mills. Impact method, you use a hammer mill or a vibration mill. Attrition method, you have this roller mills, which is usually for suspension paste and ointments. The ball mill does size reduction with a combination of impact and attrition, where you have to see, in this one, you have to see what velocity you need for size reduction to occur. If it's too high, then with this centrifugal force, there'll be no size reduction. Then you have the fluid energy milling, where the the air that is injected with high pressure usually will collide with each other, causing size reduction. And when you want to select a method, you have to see what the degree of size reduction and the cost of size reduction. And it's usually the smaller the size that you want, the higher the cost of the equipment. Yeah. That's it. And there's one more presentation that we have to do. We can take a 5-minute break and then I finish with the last presentation.
That's fine. That's fine. We can reconvene at 6:55.
Yeah, that's okay. Is the last thing is mixing. So of course, when we're talking about mixing, you have to ensure that there's even distribution of the active compound, there's even appearance, especially when you have like different colored excipients or APIs, and ensure that the dosage form releases the drug at the correct site and at a desired rate. So there are 3 types of mixtures. First one is the positive mixture, where when you mix it, it's spontaneous and irreversible, and it's caused by diffusion, and it approaches like a perfect mix. We'll talk about perfect mix just in a bit. So positive mixtures, it occurs spontaneously and irreversibly by diffusion, and it tends to approach a perfect mix. When we're talking about negative mixtures, the components usually like separate out. So what you need to do to avoid this separating, you have to constantly like mix it. So when we're talking about like calamine lotion, where the dispersion of the solid. So this is usually for like emulsions, creams, and viscous suspensions. So you have to constantly keep on stirring them. Like when we're talking about like suspensions, we have to mix it before use every time. So it usually separates out. Then the next one is neutral mixtures, where it either does not mix spontaneously or it segregates spontaneously. Oh, sorry, not segregate. It doesn't, there's no tendency to like, to mix properly, or once it has been mixed, it will not segregate. So this is an example of like mixed powders, pastes, or ointments. You guys can hear me?
Loud and clear, Daktari. Loud and clear.
Okay. So types of mixtures: positive mixture, where it mixes spontaneously and irreversibly, approaches a perfect mix. Negative mixtures, it separates out, and you usually need to like keep on stirring it. An example of this is emulsions, creams, or mostly this suspensions. Neutral mixtures, where it has no tendency to mix spontaneously, or once it has been mixed, it will not segregate. So at the start, it will not mix spontaneously, but once it has mixed properly, it will not segregate. An example of this, the mixed powders, pastes, and the ointments. So when we're talking about the perfect mix, when you start with 2, like this is, for example, this is the API and this is the excipient. You start out as this. When you want to approach a perfect mix, it would be something like this. But we don't get that in practical. It's usually something called a random mix. So that's for perfect mix and a random mix that we usually get in practice. Now, when we're talking about scale of scrutiny, it's this is the weight over volume of a dosage unit. So it is the amount of material within which the quality of mixing is important. So for example, if the unit weight of a tablet is 200 mg, then you need to pick out 200 mg of the sample to be analyzed to see if mixing is adequate. Let me repeat that. Scale of scrutiny is weight over volume, and it's an important part of mixing. if the unit weight of the tablet is 200 mg, then you need to pick out 200 mg from the sample and you analyze that 200 mg to see if mixing is done properly. So over here, the scale of scrutiny is set at 200 mg. So when you take that sample of 200 mg, you need to see, when you analyze it, you have to see that there's that 200 mg of that API, for example, in that sample. Then when we're talking about number of particles, this usually increases. It depends on the sample weight, particle size, and particle density. So when the number of particles increases when the sample weight increases and when the particle size and density decrease. So when you want to increase your number of particles, you increase the sample weight and you decrease particle size and the density of the particle. Then, so when you're measuring or you're analyzing this scale of scrutiny, or you analyzing this like 200 mg, you need to see like it has like the lowest deviation. You don't need a high deviation. That means that mixing is not done properly. If it's a low deviation, then it's good. The lower the deviation, the better. So how can that problem of high deviation be reduced is by increasing the number of particles in the scale of scrutiny by reducing the particle size. But when you're reducing the particle size, now you have all these other effects of particle aggregation, and we know that the finer the particle, the more issues it will create in like tablet making. So when you're talking about evaluation of degree of mixing, you come across something called the mixing index, which compares the standard deviation of a sample at the start and the standard deviation of a fully random mix. So when talking about mixing index or M is equal to standard deviation of the random mix over standard deviation of the mix under investigation at the start. So when you plot a graph, you expect to see the standard deviation of where is that? Yeah, so you expect to see SAT to be higher compared to now the random mix. But when you keep on mixing and when you approach a random mix, SR, or the standard deviation of the random mix, will be higher. So when the mix is if the mix is sufficient, that's when SACT is equals to SR, or when M or mixing index is 1. So when we said that when this SACT is higher, then M will be lower because indirectly proportional, inversely proportional. And when you approach a random mix, SR, or standard deviation of random mix, will equal to this. That means that M will be 1. So when M is 1, that is like now your perfect mix. Well, not a perfect mix because you can't get a perfect mix, but that randomness. So when there are two basic requirements to evaluate a mixing process. One is like you take a minimum of 10 samples at different depths. If you went to the industry, I'm sure you have seen this, where they use that sampling thief to remove the powder from different depths. Then you have to see a suitable analytical technique that, like, what analytical technique that is available. Then for this one, I'm not too sure. So I'm not, unless if somebody has gone through this, they can explain. It's the standard deviation value, which I didn't really understand how it's supposed to be calculated. Yeah, okay. So we can just go to mixing. When you have to mix powders, there are three types of mechanism. First one is the convective mixing, where there's a transfer of large groups of particles over from one part of the powder bed to another. So it's just like large groups of particles, not that you have individual particles being mixed. That is convective. Then when you're talking about shear mixing, it's when occurs when a layer of particle moves over another layer, so that you can achieve mixing by that. And the third one is the diffuse mixing, now where a true random mix is you kind of get a true random mix, because now this is a movement of individual particles. So the first one, there are three types of mechanisms for powder mixing. First one, convective mixing, where large particles are transferred from one part of the particle bed to another part of the powder bed. Second is the shear mixing, where now the mixing is from like layers, occurs when one layer moves or flows over another layer. Second is diffuse mixing, where now the individual particles are being moved and mixing is achieved. A true random mix is achieved. Yeah, so that is for mixing. Then when we talk about demixing, is when now the segregation of the powder occurs because of like effects of size or density or shape. So when we're talking about so one way where segregation is likely to occur is when that powder bed is subjected to vibration and it increases the flowability of those particles. So segregation is the segregation of the powder. Yeah, so segregation occurs because of particle size effects. So for particle size effects, we have two types of segregation, the percolation and the trajectory. So what happens in percolation is that smaller particles usually fall through larger ones because of that void. So they occupy those voids between the larger particles, and therefore percolation segregation occurs. With trajectory, what happens is that larger particles have a higher kinetic energy, so they will move to greater distances than the smaller particles. So that's trajectory. So segregation, the first one that that leads to segregation, is particle size effects. For percolation segregation, it's the smaller particles that now fill the voids between the larger particles. And trajectory segregation, where the larger particles have a higher kinetic energy and therefore they move to greater distances. So then another type of segregation is the elutration or dusting out. So I think everybody has seen this or experienced this, is that when you pour like a powder, you'll see now the finer particles, they will not like settle, they will actually be blown upwards. So once you have like, when you pour the powder and the mixture is stopped. Okay, wait, let me start that again. When you're mixing something and it has these finer particles, they'll be blown upwards. But when you stop this mixture, it will sediment on top of the coarser particles now, that is elutration segregation or dusting out. Then the second effect is particle density effects. The more dense a particle, the more dense the particle, it will have more, like it will move more downwards compared to like now the smaller particles. So if it's more dense, it will move more like downwards. That's similar to like sedimentation. If it's a larger particle or more dense, it will move downwards easily. But what happens is that the particle size and the density usually cancel each other out if the larger particles are dense. Then you talk about shape of the particle. Spherical shapes have like more flowability and they can be easily segregated than non-spherical particles. What happens with non-spherical particles, what happens with non-spherical particles is that they can interlock with each other, and it's not easy to segregate those out. So if it's more spherical, then they have like a greater flowability and they can easily be mixed, but they segregate out easily. Yeah, so when you're talking about segregation, what affects is the particle size effects. First is vibration. When you subject the powder bed to vibration, the particles will have a greater flowability and segregation will occur. When you talk about particle size effects, percolation and trajectory. And elutration segregation also, or dusting out. Second is particle density effects, where the more dense particles have a tendency to move downwards, but it usually cancels out with this particle size. And the last one is particle shape effects, where spherical particles are easily segregated compared to non-spherical, because non-spherical particles usually like interlock. So it reduces the tendency to segregate once it has been mixed. So during the initial stages of the process of mixing, when you're mixing, there'll be the rate of mixing will be greater than the rate of demixing. Then after a while, demixing will occur until like an equilibrium has been reached between mixing and demixing. So let me repeat that again. During the initial stages, rate of mixing is higher compared to demixing. Then over a period of time, demixing will occur until now equilibrium has reached between demixing and mixing. Then in formulation, the segregation problem can be addressed by a number of approaches. You have to look at various things now, like the particle size, like the selection of the particle size fractions, the excipients, like the size of the excipients, not just the API, the milling, like how the milling components of components to reduce the particle size, control crystallization, selection of excipients, granulation of the powder, reduce the extent to which the powder bed is subjected to vibration, using filling machine hopper, use of filling machine hoppers designed so that the powder residence time is minimized, and use of equipment where several operations can be carried out without transferring the mix. Yeah, so there are various factors for that. And then just go to the next thing. Yeah, so there for powder mixing equipment, there are different equipments that are used, like this tumbling blenders, which are different, like there are different types of that, such as the double cone, twin shell, cube Y cone, and the drum mixers. In most industries, the mixing is usually done by an intermediate intermediate bulk containers. So the mixing containers, I'm not sure if everybody saw this when they went to industry. The mixing containers are usually mounted so that they can be rotated on their axis. So when it's operated at a correct speed, the tumbling action is achieved. So when shear mixing occurs as the velocity gradient is produced, the top layer on the powder bed will have the greatest velocity compared to now the lower levels. Yeah, so the layer that is directly in contact with the powder bed will have a lower velocity compared to the one that's at the top of the powder bed, the layer that is on top of the powder bed. So because of that, when the bed tumbles, it dilates, allowing the particles to move downwards under gravity. So there's like diffusive mixing that occurs, where we say that they're individual particles mixing, and that gives like now the true random mix. Then you have the high-speed granulators, where to prevent segregation, because when you're moving this powder from like one equipment to the other, it can cause vibration and segregation can occur. You usually want one equipment that has like more than one function, like this mixer granulator which can do both mixing and granulation. So once with this mixer granulators is what happens is, when you have to mix the powder first, once you've achieved that true random mix, is when you add now the granulating agent in the same equipment, which will now do that process of granulation. Then you have the fluidized bed mixers. I think with fluidized, yeah, for fluidized bed mixers is mostly drying of the granules or coating it with multiple particulates. Then you have the agitator mixers. In agitator mixers, it depends on the motion of the paddle through the product. Like we've seen in like this baking shows, like you use that paddle, or when you're baking, you use that thing that you, anyway. Yeah. I think that's all that we need to do. Like I've picked out the main points. So let's just go through this one more time. Okay, so when we're talking about mixtures, you have three types of mixtures. Positive mixture, where it mixes spontaneously, irreversibly, approaches a perfect mix. Negative mixtures tend to separate out. Neutral mixtures, at the start, they don't want to mix, but once they have mixed, they will not separate. For negative, an example is calamine lotion, or mostly this suspensions. Neutral mixtures, an example is this mixed powders, paste, and ointments. Mixing process, the perfect mix would be something like this, but in practice you don't get to see that. You get to see a random mix. Scale of scrutiny is weight over volume, and it is the amount of material within which the quality of mixing is important. The number of particles depends on weight, size, and particle density. And if you want to increase the number of particles, you increase the sample weight, but you decrease particle size and density. And we need the lowest deviation in the content. So this is, again, you just increase the number of particles by reducing the particle size. Then when you're talking about degree of mixing, you have the mixing index, which when is equal to 1, that means it's sufficiently random and you can stop your mixing at that time. To check that mixing is done properly, usually take minimum of 10 samples at different depths from the mixture using a sampling sieve, and you see like a suitable analytical technique that you can use to analyze the sample. This one I'm not too sure. Then when you're mixing powders, there are 3 main mechanisms, where the first one is convective, where there's transfer of large groups from one part of the powder bed to another part. Shear mixing is the second one, where one layer moves or flows over another layer. Diffusive mixing is now movement of individual particles to achieve like a true random mix, which is now like the best mixing. Then when you're talking about demixing or segregation, it is affected by vibration. When you move particles from one place to the other, it is subjected to vibration, and that is not, we don't want that. Then it's affected by particle size effects, where you have percolation segregation, where smaller particles go through or fall into the voids between the larger particles, and trajectory segregation, where larger particles have a greater kinetic energy, so they move further. And elutrition segregation is dusting out, where once you have stopped the mixture, the finer particles that are in the air, they will settle down and they will form a layer on top of the coarse particles. Then particle density effect, where if it's more dense, it will move more downwards, but it's usually cancelled out by the size of the particle. And the last one is particle shape effects, where spherical particles tend to segregate out more compared to irregular particles, where irregular particles usually interlock with each other. Then at the start of the process for mixing, rate of mixing is more compared to demixing. Then over time, demixing will occur until an equilibrium situation has reached between demixing and mixing. There are various things that you need to look for when you're looking at segregation. The size of the particle, again, whatever we've discussed up above over here. Yeah, the different types of equipment that are used for mixing. Usually these tumbling mixtures or blenders, and there are different types: the double cone, twin shell, cube, Y-cone, or drum. And when the shear mixing occurs as the velocity is produced, so the layer that is at the top completely will have a greater velocity compared to the one that is at the bottom. And because of this, it allows the particles to move downwards under gravity, and diffusive mixing occurs. Then you have the high-speed mixer granulators, where mixing and granulation can be done in the same equipment. Fluidized bed mixtures, where it's mostly for drying of the granules and coating of multiple particulates. Agitator mixtures, where they use a paddle to mix now the powder. And usually there are these dead spots in this agitator mixtures. That is one of the limitations for that. And I think that's it. Any questions?
No questions, Cyril, no questions. But it was very nice. You've really summarized it well, especially on those types of mixing and the what types of mixing, and because I've seen that being tested, and the optimization of rate, that it shouldn't be too fast and it shouldn't be too slow, just a balance between the two.
Yes.
Yeah.
So I think that's it for
Yeah.
my part. So we're done with particles and powder technology.
Thank you. Let's see. Let me just pause the recording.
Find bioavailability. Okay. Yeah. So I don't know. Are we even supposed to do this bioequivalence as well? Ama that's somebody else's.
Na ni yako biopharmaceutics yote ni yako.
Okay. So sikuwa nime prepare for bioequivalence, but anyway, let's see. So for bioequivalence, it's when you have two drugs, similar dosage forms of it's the same drug with similar dosage forms that reach the blood circulation at the same time and extent.
Endelea.
Kuna mtu ako na simpler definition of bioequivalence. So, okay, anyway, I'll get back to it. Sai sikuwa nime prepare for bioequivalence, so acha ni do bioavailability because time pia inasonga. So, we have two main factors that will affect bioavailability. We have the pharmaceutical factors and the pharmacological factors. So the pharmaceutical factors are majorly related to the drug itself, and then the pharmacological, it's the body system and the body majorly. So kwa pharmaceutical factors, we have factors such as, let me just open this other nini. Oh my God, zote ni za ads. As we skip the ads, some of the properties, kama vile Chai had discussed about solubility and dissolution. So that's a pharmaceutical factor that's going to affect bioavailability, because if a drug is more soluble, then itakuwa absorbed faster kwa the bloodstream, ama itakuwa absorbed faster, so its bioavailability will be higher than a drug which is not easily absorbed into the bloodstream. So also there's the factor for it's on mechanism of drug absorption. Oh my God.
The ads, man.
Yeah, so, you can go through those mechanisms of absorption later. Kidogo. Yeah. So here we have the pharmaceutical factors, the drug solubility, the particle size, the polymorphism, salt form of the drug, ionization, lipophilicity, drug stability. Yeah. So there's also patient-related factors. Okay. Okay, so anyway, let me just give you a summary according to the textbook. So there was solid state properties. So we know that crystalline, most drugs zenye ziko in solid form, they are crystalline in nature, and the ones that have weaker bonds, they are going to dissolve faster than the ones that have stronger bonds in the crystalline nature. Sijui niite lattice. Yeah. Or the crystal nature of the drug. And then those drugs that are amorphous, those that lack the crystalline form, will dissolve faster. So a drug that will generally dissolve faster will have a higher bioavailability than a drug that will not dissolve faster, or a drug that will dissolve much slower. So I think that solid state and solubility and dissolution I've explained. So for particle size, the smaller the size of the particle, the higher the solubility, and therefore the higher the bioavailability of that particular drug. For the state of ionization, we know that if it's more ionized, or if it's been ionized, it's more likely to be to dissolve faster. So if the drug has is more ionizable at body pH, especially if you're taking a tablet and it's ionized at let's say like the stomach pH, it is going to be absorbed much more faster, and the bioavailability to reach circulation much faster. So that's for ionization, because like most drugs, they are weak bases or weak acids. So zinaweza kuwa ionized depending on the pH of the medium in which the drug is being, like, say if it's going to be absorbed in the stomach, we expect it to be ionized at acidic pH. If it's going to be absorbed in the ileum, we expect it to be at basic pH. Kidogo tu.
I think after Jambi is done, I can take you through pharmaceutical microbio, just the introduction.
Poa. Nice, nice.
But tutaenda mbio juu ni vitu obvious. It's about 270 pages. It should be done in like half an hour.
Okay. Kuna mtu alikuwa anaendeleza ama?
Ni wewe tu.
Okay. Sawa. Yeah. So tulikuwa tumefika kwa ionization. I think we've understood. So if a drug is more ionized, it means it's more polar. Sorry.
Okay, so if I can finish up on the explanation of the ionization in relation to bioavailability. Yeah, so here she was talking about how we've seen that the common form of drugs is they usually either as weak bases or weak acids. So here the weak acids, dependent on whichever state that they are in, the drugs that are more ionized will penetrate much better in bodily fluids, hence better absorption and distribution. And drugs which are unionized will penetrate much better in the membranes. Yeah, so that's it on ionization. I don't know if anyone is willing to continue with the presentation, because she sent the slide. She's saying she won't be available to continue.
Yeah, I think Jambi is at work. If you guys will allow me, I can take you through the Nini. Can take you through pharmaceutical microbiome. Is that okay?
Yeah, that's fine, that's fine. So Jambi amefika wapi ndio kukue continuity when
Ionization.
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 hapo kwa okay.
So guys, if you can stop her screen sharing, and I'll share mine. So I hope everyone can see and can hear me clearly.
Yeah.
Okay, let's go. So we will review the biology of microorganisms. These are bacteria and viruses, and also what are they called? Just give me a sec. Just give me a sec. Okay, let's carry on. So we'll start with bacteria. So these are prokaryotes. They have no nucleus or organelle bound in membrane. And we know the difference between eukaryotes and prokaryotes. I think it has been touched on before. So DNA is a single circular chromosomes. It contains no histones associated with DNA. Then cell walls are made of peptidoglycan, which is a polymer of NAG and NAM. Who remembers what NAG and NAM was? If you do, just add it to the chat. They are cross-linked with polypeptide chain. Then they contain smaller ribosome units, and they divide by binary fission, which is meiosis. So basically, these are typical bacteria. I'm gonna zoom it in for you guys. So we have the glycocalyx, which is a coating or layer of molecules external to the cell wall. And we have the chromosome or nucleoid, which is the site where the large DNA molecule is condensed into a packet. And we know the function of DNA is basically the primary role is controlling the cell and also in cell division. The pilus, these are elongated hollow appendages that are used to transfer DNA to other cells and in cell adhesion. So cell adhesion is the attachment to other cells, which basically make the bacteria, cause things like infections. Then we have the mesosome, which is an extension of the cell membrane that holds into the cytoplasm and increases the surface area. Oh, it folds, not holds. Then there's a flagellum, which is a mode of transport for the bacteria from one place to another. Then we have the fimbriae, which are fine hair-like bristles from the cell surface that help in adhesion to other cells. So fimbriae and the pilus work hand in hand. Then there's a granule, which stores nutrients to support, to support the cell and also provide stuff like energy when the cell needs to, undergo its basic functions. Then we have the cell wall, which is a semi-rigid casing that provides structural support and shape for the cell. Then we have the cell membrane, which is a thin sheet of lipid and protein that surrounds the cytoplasm and controls flow of materials in and out of the cell pool. And finally, we have ribosomes, which are tiny particles composed of protein that are the sites of protein synthesis. Okay, so this is another diagram of the same. Then this is a macro photo of how a basic bacteria looks like. Then this is a super simplified photo of bacteria. Yeah, and all these are just photos. And now here we have the binary fission, which shows how the cell divides into two, form what? Sister cells. Yeah, so we have the prokaryotic chromosome, plasma membrane, and cell wall, which are the basic units of the bacteria. Then they divide into two, and as we said, this is through meiosis. So if you can go through meiosis, you'll get a better understanding of that. So eukaryotes have a true nucleus and are membrane bound. Then DNA is definitely membrane bound inside the nucleus. The nuclear membrane is a double membrane. Okay. So the cell wall is basically made up of cellulose or chitin. Then I think everything else you can read. Then these are typical eukaryotes. Now these are cells like found in plants and human mammals, basically. Okay, and we can go on. All these are photos of what the bacteria is. Then now, okay, so you guys may need to read about the lipoprotein, peptidoglycans, the phospholipids, cytoplasmic membrane, and the porins. That's about it. So peptidoglycan is also known as murine, and peptidoglycan can be seen here. I don't know if I selected it. Okay, peptidoglycan can be seen here. Okay. Yeah, basically all these other things, you can just read and go through them slowly. Now, there are two types of bacteria. There's a classification of bacteria where we have the gram-positive and the gram-negative. How do we find out if bacteria is gram-positive or gram-negative? We do a lab test where we fixate the bacteria onto a slide. If you guys remember, we did this, I think, in second year. We did this in second year. So you have your gram-positive, you have your bacteria, you fix it on a slide, then you apply crystal violet, which is a solution. Then you treat it with iodine. Then you wash it. Once you wash it with an alcohol-based solution, if it's gram-positive, it will retain the purple color of the crystal violet. If it's gram-negative, it will decolorize. Then you counterstain with safranin. So for gram-positive, the purple color is retained throughout all the treatments. But in gram-negative bacteria, you find that the bacteria changes color from purple and to safranin is kind of reddish pinkish. So the bacteria will change color to a red hue or a pink hue. Okay, so this is basically how it looks like, how the process flows. Then what is the difference between a gram-negative and a gram-positive? So a gram-negative cell envelope is complicated than a gram-positive because the gram-positive contains lipid, phospholipid, lipid A, periplasmic space with various proteins. But for the gram-negative, they only have the cytoplasmic membrane. They don't have the outer membrane. I hope that is understood. So cell shape is determined by genetic character of the organism. So the bacteria can also be classified based on its shape. And let's just keep all this and go to the different shapes. So there are bacilli, which are rods, kind of. Then, bacilli are rod-shaped. Then there's a coccus, which is round-shaped. Then there's a staphylococcus, which occur in round-shaped, but in clusters. Then we have the diplococcus, which are round-shaped, but always appear as two. They always appear as two bacteria clustered together. Then we have the spirilla, which is a corkscrew shape. I know some of you may have indulged in wine drinking. If you end up taking expensive wine, you know the cork, you can't just pull it out, you have to use a corkscrew. So the corkscrew shape is what is also defined as spirochete in bacteria. Then F, the last one, we have the vibrio, which is this here. It's comma-shaped. And this can be seen in bacteria such as cholerae. So spherical is coccus or cocci. Then rods, we have bacilli or bacillus. Then spiral bacteria. And you can see the photos of all these bacteria. Give me a second. So moving on swiftly, the spherical bacteria, we have several. We have diplococci, which appear as two, and they divide in one plane. Then we have the streptococci, which is a cluster, and they divide in one plane severally. Then we have tetrad that is like a diplococcus, but divide in two planes. So they'll form a cluster of 4. Then we have Sarcinae, which divide in 3 planes. That's the X, Y, and Z axis. So they'll form like a cubical kind of shape. Then we have Staphylococci. They divide in multiple planes and remain as a cluster. And the multiple planes are not well defined. For the rod-shaped, we have single diplobacilli, streptobacilli, and coccobacilli. So coccobacilli looks more circular, but it's still a rod-shaped bacteria. Then streptobacilli elongate along a single line, which is the y-axis. Then diplococcus are usually a pair. Okay. Then the spiral shaped is basically spiral that's like drawing. A spiral shaped would be like a spring. It will look like a spring, yeah. Then the spirochete we said is a corkscrew, and then we have finally the vibrio. But we also have Helicobacterium, which is a curved rod shape. Okay. Yeah, now on to viruses. So viruses are obligate intracellular parasites and only demonstrate characteristics of life when inside a host cell. So when they're outside a host cell, they're basically inactive. But once they get into a host cell, which can be bacteria, an animal, or a plant, they start showing characteristics of life. What are characteristics of life? One, they eat, drink, poop, reproduce, die. That's basically what characteristics of life mean. So for viruses, they're inert, outside the host cell, they're inert, no enzymes or any other activity. But when they're inside the host cell, they have viral nucleic DNA or RNA and take over the cell and directs the cell to produce new virus particles. So what are they? Viruses, if we talk about them in IT lingo, viruses affect computers by inserting themselves inside the software and tell the software to reproduce copies of itself and affect the basic function of the software. So basically, it's exactly what happens when viruses affect humans or animals or plants. So when they enter our bodies, they enter our cells, they start feeding off of us, and they also reproduce and begin controlling how our cells work. That's why you find maybe someone may get lowered immunity, someone may get other symptoms. For example, if you have like influenza, you start getting sick, you start getting fevers, things of the sort. So a basic virus particle is known as a virion, and this is an intact and infective virus particle. So the components of this is a nucleic acid, which is a DNA or RNA, protein coat, which is a capsid made of individual protein subunits. You can see the capsid around here, and the capsid encompasses some form of DNA or RNA. Some may have an outer envelope, a membrane derived from the host cell. The envelope can have spikes of protein, H&N spikes of influenza, that aid in attachment and make them sensitive to chemical actions of disinfectants. So types of viruses, the morphology, we have the shape and the structure. So we have the helical shape, and you can see that here. And then we have polyhedral, which is adenopolio, for example, enveloped, which is a flu, and complex bacteriophage. So this is a complex bacteriophage viruses. So we have different types of viruses. We have the Ebola virus, we have the polio, we have the influenza A, which is enveloped, and you can see the spikes around here. Then we have the bacteriophage or complex. So this is an example of a bacteriophage or a complex virus. So this is complicated and boring, and we'll leave it for the ones with a higher pay grade. Yeah, so if you get a question like this, it's not in your pay grade. Thank you. So viruses, cultivation, you need living cells. So the living cells are Spider-Man. Spider-Man.
Yeah.
The famous lady who gave us the cells that never die. What's her name? Or anyone who remembers her name. The cells that never die. So they're called HeLa cells, for those who don't remember. They were donated, they were obtained illegally from Henrietta. So HeLa cells are basically what we use to cultivate bacteria and viruses, because they give a good view of what the cell is like. But I mean, of what the virus multiplication process, infection process is like. So we can also use living hosts. We can use living hosts include things like rats, bunnies, dogs, cats, and horses. We can also use tissue cultures, embryonated eggs, and bacterial cultures. So bacteria are grown as lawn, and viruses are in the clear zones, as you can see here. in this diagram. Okay, so this is a basic life cycle. So the first step in multiplication, you absorb the bacteria onto a living cell, then the virus. Then the virus will penetrate and there will be a viral chromosome into the host cell. As you can see, this tiny thing. Then there's synthesis of early proteins. Then there's replication of viral DNA within the cell. Then there will also be multiplication of viral chromosome. Then we have synthesis of late proteins and assembly. Then the viral chromosomes are surrounded by newly synthesized protective coats. As you can see here, they've formed the two coats on the top of their head. Then lysis. Lysis is a cell. Lysis is of the host cell. So when there's lysis of the host cell, there's a release of new virus particle into the surrounding area, which can be into the what is it, the extracellular matrix? If it's in a human, it's in the blood, things like that. So viral replication in life cycle of bacteria virus, we've gone through that, and we can just skip this. We can also skip this. Okay, we can skip all this. Then we can go to fungi. That's the name I was trying to remember. So fungi are eukaryotes. Nearly all are multicellular, distinguished from other kingdoms by the form of nutrition, their structural organization, their growth, and reproduction. So absorptive nutrition enables fungi to live as decomposers and symbionts. So you'll find a lot of fungi growing next to dead bodies, dead trees, dead animals. You'll also find them in the floor of big rainforests. So fungi are heterotrophs that acquire nutrients by absorption. They secrete hydrolytic enzymes and acids to decompose complex molecules into simpler ones that can be absorbed. If you guys watch Murder Mysteries, you'll find that most people who are scientific enough, when they bury dead people, they tend to spread spores of fungi, so that it aids in decomposition of the bodies of the deceased. And this, because of how they secrete hydrolytic enzymes, they tend to decompose the bodies really fast. And after a few years, let's say 2-3 years, there will be no what do you call it? There'll be no skin and there'll be no muscle found on these bodies. And chances are the only thing you'll find are bones. So specialized into 3 main types. There are saprobes, which absorb nutrients from dead organic material, parasitic fungi, which absorb nutrients from cells of living hosts, and some are pathogenic. For example, athlete's foot is one of the conditions that you find parasitic fungi in. Then there's mutualistic fungi, which absorb nutrients from a host, but reciprocate to benefit the host. So basically, this is how it looks like. There's a reproductive structure that contains the spore-producing structures. Then there's a hyphae that flows from point of contact with the dead or living organism. So it goes in to the dead or living organism and out into the fungi. Then whatever absorb the root in quote structure of the fungi is called mycelium. So the basic structural unit of fungal vegetative body is a hypha, or in multiple hyphae. Except for yeast, hyphae are organized around and within a food source composed of tubular walls containing chitin. We've also seen chitin again in bacteria. So they provide an enormous surface area. So fungal hyphae may be septate or aseptate. So hyphae of septate fungi are divided into cells by cross walls called septa, which you can see here. And then we have hyphae of aseptate, lack cross walls, and they're coenocytic. Okay. Then parasitic fungi have a modified hyphae called Haustoria, which penetrate the host tissue but remain outside the cell membrane. Excuse me. So you can see this is a fungal hyphae on Haustoria, which has embedded itself into a living organism, which in this case is a plant cell. So fungi reproduce by releasing spores that are produced either sexually or asexually. They're usually unicellular, haploid, and of various shapes and sizes. They can be produced by meiosis or by mitosis. In favorable conditions, fungi generally clone themselves by producing enormous numbers of spores asexually. For many fungi, sexual reproduction only occurs as contingency. These result in greater genetic diversity. Spores may be carried by wind or water and germinate in moist places with appropriate substrata. Okay. So I can go through this. It's not very important, but it may be important. Phylogeny of fungi. So you can go through these different life cycles of different types of fungi. Then we have molds. Molds is important because as we're discussing aseptic techniques, we know molds form especially in rooms that are dark, rooms that are very moist. So you may find fungi growing even on your lab tests. You may find it growing on your samples. You may find it growing within the room that you're operating in, especially if it was not built according to standard guidelines. So it's important to also learn about molds. So they're rapidly growing fungus with no sexual stages and may develop into a sexual fungus, producing zygosporangia, ascocarps, and basidiocarps. Molds with no known sexual stage are known as deuteromycota or imperfect fungi. For example, penicillium, or the flavor for blue cheeses. If you guys are lovers of cheeses like me, you'll know that mold slaps for these. Next, we will talk about yeast. So these are unicellular, reproduce asexually by budding or sexually by producing Asi or Basidia. And we said when fungi reproduce sexually, it's mostly because of contingency. They have no other option. So Saccharomyces cerevisiae is most important domesticated fungi, and it's used basically for baking and brewing, and it's also a model organism. And Saccharomyces are also given as a prebiotic. Probiotic. They're given as a probiotic for people who've been on antibiotics for a long time, or maybe if you're using a high dosage of antibiotics, it may ruin your stomach normal flora. So you're given Saccharomyces to improve your gut flora. So it can cause problems such as rhodotorula, which is usually found in shower curtains. You know that slime, the slime that you find in your shower curtains. That's rhodotorula. And then Candida, which is thrush. Candida is in many shapes, forms and sizes. It can be found in the reproductive organs, especially for ladies, where they get things like UTIs. It may also be found in the mouth for people maybe who are living with retroviral disease. So it can be problematic, and we need to learn how to control it adequately. Then we have lichens. Lichens mostly found in plants. They provide fungus with food, and they usually fix nitrogen. The fungus provides good environment for growth. They have hyphal mass absorbs. They have hyphal, where the hyphal mass absorbs mineral and protects the algae. So you see how it's a symbiotic relationship here. Lichens are mostly found in plants. So the reproduction, you can go through it by yourself, but the symbiotic, I'll just read for you the simplified one. So symbiotic units reproduce asexually by fragmentation of parent, where it breaks off and it breaks off the main lichen and starts developing by itself. Then there's formation of Soridia, which are small clusters of hyphae with embedded algae. So in this case, you find that the Soridia has embedded into a piece of algae, and because of the symbiosis, it continues to continually grow and continues its basic life functions symbiotically with the algae. Then we have mycorrhizae. Mycorrhizae. So this is a specific mutualistic association of plant roots and fungi. Fungi increase absorptive surface of roots and exchange soil minerals. It is found in 95% of vascular plants, and it's necessary for optimal plant growth, especially in things like potatoes, because there are those they require a lot of nitrogen, and mycorrhizae are nitrogen fixing fungi. Then ecological impacts on fungi as decomposers. Some fungi are pathogens, as I said, athlete's foot and things like ringworms. And plants particularly are susceptible to fungi disease. Maybe, for example, the Dutch elm disease. Ergot, as we had discussed like a week or two prior, we know the ergot derivatives and whatnot, they affect cereal crops. They cause gangrene, hallucinations, and St. Anthony's fire. So you guys can read on that. Many animals, including humans, eat fungi. In US, mushroom consumption restricted to Agaricus, because there are so many different kind of mushrooms. And as Chai can effectively tell you, there are some types of mushrooms he consumes that may make him hallucinate and think shots are the best option for the world. So we eat a range of cultivated and wild mushrooms. Truffles are underground as cocups of mycelia that are mycorrhizal on tree roots. Yeah, now we get into the aseptic techniques and safe handling of hazardous agents. So those are the learning objectives. So infection control. So what is germ theory of disease? The identification of microorganisms as a cause of infectious disease is a surprisingly recent development. Pasteur was the originator of the theory. In one experiment, he successfully immunized chickens against chicken cholera. So we know Pasteur for his pasteurization methods, which is superheating liquids, specifically things like milk, so that they become sterile and kill off things like bacteria and fungi that may be found when obtaining the milk in this case. So microorganisms and disease. Since the days of Pasteur, thousands of pathogenic or disease-causing microorganisms have been identified. These are not limited to bacteria. These include, but are not limited to bacteria, fungi, viruses, and protozoa. So asepsis sterilization. The preparation of parenteral products, especially those for IV administration, requires special aseptic techniques, because you don't want to go treating someone, then you give them another disease, maybe from a bacteria that wasn't killed off when preparing the drug that you administer to this patient. So they require special aseptic techniques, equipment, and procedures to minimize contamination. Various types of sterilization are available to kill microorganisms from medical instruments, devices, and surfaces. Hello, guys. Are you guys still there? Hello. No one is around? Oh, no. Okay.
Patricia here.
Various types. Good. That's a motivation. So what is contamination? This is an accidental exposure that require immediate treatment, cleanup, and reporting to the supervisor and completion of an incident report. Contamination can occur in a pharmacy by 3 primary means. So there's touch, where you touch something maybe without washing or sterilizing your hands, the air that is circulating within the room, and water that is used to prepare the water that is used to prepare the solutions or the medication or the infusions. So you need to effectively control these three aspects to avoid contamination. So aseptic technique under the USP chapter 797, we define it as the technique used for preparation of parenteral admixtures, combinations of fluids and of medications or nutrients which are administered using bolus, that is push or other intravenous methods. So the aseptic technique aims to prevent pathogenic organisms in sufficient quantity to cause infection from being introduced to susceptible sites by hands, surfaces, and equipment. It protects the patient during invasive clinical procedures by employing infection control measures that minimize, as far as practically possible, the presence of pathogenic microorganisms. So these may be sterilizing surgical rooms or theaters, using well-sterilized equipment, ensuring the medication is prepared in a safe manner, the surgeon and the nurses helping him, ensuring they wash their hands, ensuring their HEPA filters that circulate clean air into the theater or operation room. Basically, those are some simple examples of why the aseptic technique is important. So the quality control sample concentration is determined from standard curve, and if the result is in the range given by the kit manufacturer, the results could be accepted. Hey, it happened. So why practice it? Aseptic technique reduces the risk of healthcare-associated infections. It is required for all invasive procedure, and it has been shown to significantly improve the practice of clinicians performing procedures and reduce the risk of infection. Healthcare-associated infections, which is HAI, are infections acquired in healthcare facilities and infections that occur as a result of healthcare interventions. HAIs are caused by transfer of pathogens to a patient during healthcare intervention, and they may manifest after people leave the healthcare facility. And this happens a lot, but with the recent development of aseptic techniques, the number is steadily going down. So preventing infections using aseptic technique, the correct aseptic technique prevents contamination and transfer of pathogens from hands, surfaces, and equipment to the patient. It requires key parts and key sites to be identified and protected at all times. Key parts must only come into contact with other key parts and/or key sites. So preventing infections using the aseptic technique, there are several key infection control components to consider when performing any invasive procedure. That is environmental control, hand hygiene, PPE selection, aseptic field management, non-touch technique, and sequencing. So for environmental control, prior to aseptic procedures, clinicians must ensure there are no avoidable nearby environmental risk factors. These might include bed making, patients using commodes, waste management, cleaning of the nearby environment, and patient bed curtains across work area to prevent transmission from one patient to another patient. And as you may find, most hospitals don't use cloth material, or if it's a cloth material, the inside is lined using easily sterilizable plastic layer. Just give me a minute, guys. Okay, so hand hygiene, effective hand hygiene is an essential component of aseptic technique, and it's dependent on the procedure about to be performed. Either routine or surgical hand hygiene is required. Routine is where you just basically wash your hands in the different steps that we were taught and were examined for, to be able to complete our pharmacy course. And then there's a surgical hand hygiene where it's a more advanced hand washing procedure that involves washing your entire hand, removing things like bracelets and watches, and you wash your hands all the way up to your elbow. So the routine hand hygiene refers to the use of soap solution and water or an alcohol-based hand rub or sanitizer. Surgical hand scrub requires the use of an approved antimicrobial skin cleanser or waterless hand rub formulation. Yeah, so this is different ways to wash your hands, how to apply sanitizer, washing your hands using soap, surgical hand hygiene. Now, PPE. So PPE, we can use things like gloves. If it's necessary to touch key parts or key sites directly, we use sterile gloves. Sterile gloves don't come in pack of 50s. Sterile gloves packed individually in pairs. And they must be used to minimize the risk of contamination. If the key parts or key sites are not touched directly, non-sterile gloves may be necessary to protect the clinician from blood or body fluids or exposure to toxic drugs during administration. Gloves do not replace the need for hand hygiene, so make sure you wash your hands. So selection of sterile or non-sterile gloves is also dependent upon clinician competency. So you use your judgment to know, for this specific procedure, will I need sterile or non-sterile gloves? So other PPEs may be required to reduce the risk of blood and body fluid exposure to the clinician. So basically PPEs are mostly to protect the clinician and also to protect the patient from getting other infections that may be derived from the clinician. So maximum barrier precautions may be required during some procedures such as CBC insertion to reduce the risk of patient acquiring a healthcare associated HAI. Then refer to your local policy and procedures to determine if maximum barrier precautions are necessary. So PPE must be worn when compounding, no jewelry, nail polish, artificial nails, or makeup, because these things may harbor bacteria and they may fall onto the sterile equipment. They must be disposable, non-shedding, and have low permeability. Don all PPE in anti-roll. So remove all PPE before leaving sterile area. A gown may be reused during a shift and all other PPE may be disposed of. So gowns being reused during shifts, this is not in places like surgical rooms. Gowns being reused usually seen in places like when you're manufacturing, let's say education and things like that. You may don the same PPE during the same shift, but you have to ensure that after that shift, everything is after the shift, everything should be cleaned and sterilized before the next day's shift. So order of donning PPE, you start with shoe covers, then head and facial covers, face, eye masks, eye shields, aseptically wash hands, then you wear your non-shedding gown, clean hands with waterless alcohol and scrub. Then finally, the last thing you put on is your gloves. So this is how a properly worn PPE should look like. Then hand washing performed before entering direct compounding area. Under warm water, use nail cleaner and soap to clean under the nails. Wash hands and forms at least 30 seconds with soap. Wash around and between all fingers, scrub up to elbow, covering the entire area. Rinse completely and dry with lint-free paper towels or electric dryer. So why we use paper towels instead of, our regular things like cloth wipes is because the cloth wipe may be reused and people may not sufficiently have cleaned, so it may still be a source of contamination. You use paper towel because once you use it, you dispose of it. So next one, aseptic field management. Prior to commencing a procedure requiring aseptic technique, it is imperative the clinician determines the aseptic field required and how that field will need to be managed. You guys can read through this. It's not much. So we can briefly go through the non-touch technique. It's an important component of aseptic technique even when sterile gloves are used. It is well documented that hand hygiene is not always correctly performed, and that even correctly performed hand hygiene cannot always remove all pathogenic organisms. Therefore, an untouched technique is a vital component of achieving asepsis. So an untouched technique is a technique where the clinician's hands do not touch and thereby contaminate key parts and key sites. The safest way to protect a key part is to not touch it. So surgical aseptic technique, it's a technique that utilizes critical aseptic field, which is treated like a key part, and also uses full barrier precautions such as sterile gloves, sterile gowns, cap, and masks. So it achieves a safe level of asepsis for procedures that are technically complex, extended over long periods of time. Large open or multiple key sites exam may, for example, a PICC insertion. So there is a guide for standard and surgical aseptic techniques. So choosing the right technique is important, and it's essential because it prevents pathogenic microorganisms from being introduced to susceptible sites by hand surfaces and or equipment. To help you choose correctly based on your skill level, it is based on your skill level and the procedural requirements. A risk assessment can be undertaken by applying a few key considerations. Okay. So sequencing, when performing a procedure, practice must be sequenced to ensure an efficient, logical, and safe order of procedure events. So practice guidelines provide direction as to the correct order in which the preparation and completion of the procedure should be undertaken. And that's why there's always a list of what to do at what time and to what extent. So clinicians should be familiar with the sequence of these events prior to commencing the procedure to ensure preparation for the procedure is complete and to ensure adherence to aseptic technique. So to prepare for a procedure, you need to assess risk, and this involves determining the complexity of the procedure, the skill and experience and competency of the one performing the procedure, the key parts and key sites associated, whether the key parts or key sites need to be touched, and what are the appropriate infection prevention measures to protect key parts and key sites. So aseptic technique, remove jewelry, put on non-shedding coats, gowns, or overalls, which can be head and facial covers, face masks and shoe covers. Then wash your hands by scrubbing them to the elbows thoroughly with an antiseptic cleanser. Clean the laminar flow hood. This usually applies when you're preparing extemporaneous preparations. Correct me if I'm wrong. Place only essential materials under the airflow hood. Scrub again your hands, then wear your gloves. Then swab or spray needle penetration closure on vials. This is usually after you break the metallic bit, there's that rubber part where you poke with a needle. You need to sterilize it just in case. Prepare the sterile product by withdrawing the medication from the vial or ampoules and introduce it into the IV container. Then complete a quality check of the product for container integrity and leaks, solution cloudiness, particulates, color of solution, and proper preparation of the product. Then present the product, the containers and devices used, and label to a pharmacist for verification of product preparation. This will mostly find it in oncology units. Then manipulation. So avoid touching the swabbed area, uncapped needles, hub of needles, syringe tip, syringe plunger, open tip of filter or dispensing pin, uncapped tip of any tubing. Swab with 70% IPA. So if you see IPA, it's isopropyl alcohol. And what do you swab? The vial tops, the ampoule necks, top of bottles, pots of any IV or VPB bags, just to make them safe. Sorry guys, I don't know why it keeps doing this. So using needle, there are different sizes and lengths. As you know, they are called gauges. If you've operated in a hospital or a pharmacy setup, you'll know the different gauges from 18. The standard gauge is 18 to 21, but they go all the way to like what, 27. So the smaller the number, the bigger the needle. The bigger the number, the smaller the needle. So handle needle only by protective cover, and don't remove the cover until you're ready to administer whatever drug you're giving. So using vials, beat all the vials before bringing them into the hood. Discard single dose vials at end of shift. Okay. Using vials to prevent coronary pressure issues. So pressure issues is when you're trying to draw from the vial. When you're using the syringe, when you don't draw correctly and you tend to, plunge the syringe back in, the pressure builds inside the vial, and this may cause the vial to explode. So you need to be very careful about that, because it may explode in your face. So withdrawing from a vial, you dry, wash hands and gather all materials, swab rubber top with alcohol and allow to dry. Make sure needle is firmly attached to syringe. Pull plunger back to slightly less than amount needed. Then remove cap, place needle, bevel end up on center of the stopper. Hold it at a 5-degree angle and insert. Some of them you can do 45, some you do at a 90-degree angle. So gently push air from syringe into vial, then insert vial and release plunger. Remove any air bubbles by tapping on the syringe, withdraw needle and carefully recap. But for most of the procedures we do not recap our syringes, our needles, as that is not very safe. So example of straight draw, you'll just go through the photos. So to reconstitute a powdered vial, dress, wash hands and gather all materials. Swab all rubber tops with alcohol and allow to dry. Make sure a needle is firmly attached to syringe. Drop correct amount of diluent needed. Pull back plunger to clear neck, remove needle and replace with vented needle. Then add diluent to powdered vial. Gently shake or swirl to dissolve. Don't shake it too hard, as this may cause it to get bubbles and things like that. Then change back to regular needle and withdraw fluid. Remove any air bubbles, withdraw needle and recap. Then offer it to the person who's administering the drug. So removing air bubbles, pull back plunger to allow more air into the syringe. Rotate large air bubble around syringe to pick up smaller ones. Tap syringe to dislodge smaller air bubbles. Hold syringe upright, pull plunger back another 0.2 mils to clear the hub. Then push plunger up to remove air and make sure desired amount is left in the syringe. It's important to make sure the desired amount is left, because if you do too much or too little, then the drug may not be effective or it may cause unwanted side effects. So using transfer needle, these are double-ended and they're used to transfer entire contents of vial to an IV bag. So to use, swab appropriate areas, place long of in port of IV bag, place shorter end into desired vial, gently squeeze IV bag and IV fluid will fill the vial. Hold IV bag so that air floats to the top and squeeze. Air will move into vial and create pressure in vial. So opening ampules, this you will do a lot of, maybe. So dress, wash hands and gather all materials, remove fluid from neck of ampule, swab neck with alcohol, hold ampule at a 20-degree angle towards side of the hood. Using thumbs, apply pressure toward the neck, and using filter needle, withdraw the fluid, remove any air bubbles, change to regular needle before injecting in bag, then remove any air bubbles. Insert needle into additive port of the IV bag and inject. So this is how you open an ampule. You use sterile swab, or you can use cotton wool. Then you hold it at a 20-degree angle, wrap it, hold it at the neck, kind of twist it, wiggle it a bit, then break off the neck, at the neck. That's it. Then preparing a sterile dosage form, assemble the materials within the working area, clean injection surfaces with 70% IPA, place syringes with needles capped on the hood, check base solution and place next to the syringe. Then don't puncture bag when inserting the needle into the port. Keep water and potassium chloride in the hood Position injection of pot or bag toward the HEPA filter. Withdraw drug fluid with appropriate sized syringe, then properly label and inspect for cause and matter. So handling issues, and these are all safety notes. When working in the pharmacy lab, clean the room. It is important, or a clean room. It is important to wear appropriate sterile gear, including face mask, full head covering, scrubs or gown, back closure and gloves, and also shoe covers. So this is an example of a air circulator or an airflow machine. So figure 1, which shows a hooded surface that where the air flows in one direction. But in figure 2, we see one that recirculates the air to prevent whatever is being carried into flowing into the room. Rather, it's exhausted out of the room. So coughing or talking should be directed away from the hood, because as we know, our mouths are hubs for many, many different bacteria and fungi. And it's important to not talk or cough towards the hood, as this may contaminate our product. Yeah. So handling and disposal, this will go through in the next session. I hope you all enjoyed this quick presentation. I think we can end it there, guys. Jasmila.
Yes. Yeah, we can end.
I'm saying,
Thank you.
sorry?
You're saying?
I'm saying you're the host of the meeting.
I'm the host.
Yeah, I think we can end it there.
Oh, sawa sawa. Thank you, Patrick.
You're welcome. You're welcome.